Dear students, welcome to R.A.T.S. Racing Against Time for Students in Pathology. I am straight away going to the topic chemical carcinogens. A little complicated and appears boring, but we shall see how well we can make it interesting. And the importance of it is given in the enclosure. Let us go to history first. There was an occupation of chimney sweeps who developed a scrotal skin cancer, and it was Sir Percival Pott who did a simple thing as making them bathe after the work. And remarkably, there was a drop in the incidence of cancer. It was caused by this polycyclic aromatic hydrocarbons or carbon soot. And there is a gentleman who was behind that revolution. I would like you people to kindly draw this diagram. There are a few steps in the formation of a neoplasm. One is initiation, the other is promotion, and finally a progression to neoplasm or even malignancy. This particular scheme you people shall and will remember. By the way, these are the terms that we should remember. Initiators are substances that cause irreversible damage to the DNA, but they do not cause frank malignancy. An example is benzopyrene. Promoters, on the contrary, cannot induce cancer but can propagate or enhance the formation of a cancer. And the classical example is saccharin. So these particular terms and schematic diagram I would like you people to memorize. This shall form the fulcrum of our answer. What are the steps in chemical carcinogenesis? We are being constantly exposed to a lot of carcinogens. Chemicals are one such thing. And what happens is the cell is being converted into an electrophilic intermediate. It can be the DNA, it can be the RNA, etc. What does it mean? In an atom, you find that there is a neutron, a proton, and an electron. These are evenly balanced. But when there is going to be an imbalance in these, there can be a tremendous amount of energy and potential which can lead to devastating damage. And the carcinogen itself can be excreted from the human body. It causes no damage. So also this electrophilic intermediate may be inactivated and excreted. But sometimes what happens is it gets bound to the DNA, and this is called a DNA adduct. It gets bound to the DNA, and here also what happens, there are various steps in DNA repair. So it becomes a normal cell, or if the damage is too much, the cell dies. There can be a cell death. So electrophilic intermediates bound to the DNA adduct formation. But if one cycle of reproduction were to continue, then the change in the DNA becomes permanent. It becomes fixed. And that particular cell is called an initiated cell, and the entire process is called initiation. Please remember the sequence: carcinogen, electrophilic intermediates, binding to the DNA, adduct formation, and multiplication to form a permanent fixed change in the DNA. The cell becomes initiated, and the process is called initiation. This itself is not sufficient for malignancy. It will be acted upon by another agent called the promoter, and the process is called promotion. And there is an altered cellular proliferation. Finally, there is the development of a neoplasm or new growth. These are the steps in carcinogenesis. Kindly by heart it. Take notes of my class. These are the steps and the summary is given here. There are two major steps, namely initiation and promotion. In initiation, you find that there is the formation of electrophilic intermediates which bind to the DNA, formation of an adduct. One cycle of multiplication is definitely needed for this change to become fixed. And if it is acted upon by another agent, it is called promotion. There is altered differentiation and the development of a neoplasm. These are the steps in chemical carcinogenesis, and you people shall and will write this. Classification is an aid to orderly thinking. I would like you people to kindly by heart it. I have somewhat summarized it, though not meaningfully. The chemical carcinogens can be divided into major carcinogens. These again are divided into direct acting and indirect acting. Direct acting, there are some examples such as drugs, cyclophosphamide, busulfan, etc. Paradoxically, these are drugs used for the treatment of cancer, but they can produce another cancer, including a leukemia. The direct acting do not require a promoter and they can straightaway cause a neoplasm. Indirectly acting, on the contrary, they need the help of a promoter. The examples are such as given in the box. So you have got benzoanthracene, benzopyrene, beta-naphthylamine, nitrosamines, etc. And these are the instances where they are activated, and this is the malignancy that is caused by it. Please do by heart this. There are also a spectrum of other chemicals which can cause malignancy. Aflatoxin B1 caused by Aspergillus flavus resulting in hepatocellular carcinoma. Vinyl chloride of the plastic industries resulting in angiosarcoma. Arsenic, it can be topical application also which can lead to a skin cancer. Smoked fish, more common in countries like Finland, resulting in malignancy of upper GIT. Chromium, nickel, etc. They are common in the mining industries and they can cause lung cancer. Asbestosis. There are a lot of industries which are involving them and it can result in tumors such as mesothelioma. So this particular list of chemical carcinogens we shall memorize. They can be asked as MCQs. Initiation and promotion are two different steps. As we had seen earlier, first there will be an initiation. This X is supposed to be indicating initiation. In group 1, I am finding that there is a step called initiation, but there is no subsequent application, as a result of which there is no tumor developed. On the contrary, group 2 and group 3, you find that there is an initiator first, and whether there is a gap or not, subsequently there is the application of the promoters, the patient develops a tumor. On the contrary, in all the other groups, group 4 and 5 particularly, the promoters are first applied or the patient is exposed to the promoters first, followed by the initiator, there is no tumor. Or only promoters, there is no tumor. Similarly, when there is going to be a gap in the application of the promoter, there is no tumor at all. So, too many X and stars, it will be a little difficult for us to memorize. See this one. So, in initiation, there is a damage to the DNA, but if there is no promoter, there is no tumor. In this case, the promoter is applied earlier to the initiator, there is no tumor. Similarly, repeated exposure to the promoter followed by the initiator, there is no tumor. So also in this case. But in the last two steps, you find that there is an initiator which is followed by promoters. Once the damage is initiated, the cell can remember it for a very long time. That is why even when there is an application, even after a long time, there is a development of a tumor. See what is happening over here. So these two rings I would like you people to remember. The last two over here, they correspond to 2 and 3. there is a development of malignancy. This particular sequence of events, I would like you people to kindly draw. Students, please concentrate. This is an important test called the AMES test, capital AMES, or the histidine test. Now, the organism that we are using in this case is Salmonella typhimurium. Normally, this particular organism is dependent on histidine. Histidine is some kind of a medium that is used, and because of that, there will be growth. Now, what happens is, in one case, we are applying this organism onto a culture plate which is lacking histidine. But at the same time, it is being exposed to a mutagen. Mutagen means it causes some change in the bacteria, as a result of which, after incubation, there is a tremendous amount of growth of the bacteria when there is a mutagen or a chemical carcinogen that is exposed. On the contrary, when there is no mutagen that is added, you find that there is no growth or very limited growth that happens over here. Three things you will have to remember in this one. One is histidine. Histidine is supposed to be the substance which is usually important for the growth of the bacteria. The bacteria that we are going to culture is called as Salmonella, and the chemical is called the mutagen, which causes a change in the DNA. Now what happens is, once this is there, the bacteria gets changed, and even when there is a lack of this histidine, after incubation, there is a growth in the experimental plate. So this is the importance of the histidine test, which means that once there is an exposure to the chemical or chemical carcinogen, the cell undergoes a change, after which it continues to grow even if the media is going to lack histidine. Even if our body is going to lack any further stimulus, there will be the growth of the tumor cells. That is the principle of this one, and please do remember this is extremely important. Coming to this one, our favorite friend, cigarette. Robin says, we are swimming in a sea of carcinogens, and the number of carcinogens can be hundreds or even thousands. I just enumerated a few of these. You find that a lot of these are present within the cigarette and the cigarette smoke, and these can induce malignancy not only in the lung but in various other organs, surprisingly even the urinary bladder. Promoters. Promoters are substances. We had earlier seen the initiators. These are some substances such as saccharin, TPA, tetra-decanol formol acetate ester, phenobarbital, and betel nut. These are all some of the promoters. So these, when subsequently applied, can lead to a malignant transformation. And this is because of the activation of an enzyme called P450 oxygenase in the liver. So this is a warning that is given for the usage of saccharin. Saccharin is very commonly used because it is 600 times sweeter than sugar. I would like you people to kindly know these things. See what happens. This is the maize that is being stored in a granary, and it is contaminated by means of aspergillus or fungus, which produces a toxin called aflatoxin B1. And this aflatoxin B1 can lead to a cancer in the liver called hepatocellular carcinoma. And this is another strange thing that is happening. On exposure to azodice or beta-naphthylamine, these are actually inactivated in the liver. But subsequently, you find that the patient can develop a malignancy in the bladder because the inactivation is again reactivated by means of the enzyme glucuronidase. That is why the patient does not develop a liver cancer, but instead he develops a bladder cancer. This can happen in leather industries with azodice, aniline dyes, etc. Also, for preservation, we are using nitrates and nitrosamines. Smoked fish, which I had mentioned earlier, all of this can lead to malignancy of the GIT, esophagus and stomach in particular. These are some examples, and they can be asked as MCQ, or even if you answer, you score a point. Asbestosis. It is a question by itself. Asbestos fibers are of numerous types. We'll be dealing with it in detail. And in a number of industries, these are included, including the manufacturing of the shipping industry or the asbestos fibers, etc. Sheets. And they can lead to, look at this lung. The lung is being encompassed by means of a tumor, both the lungs. And that is called as mesothelioma. This can also occur in the pleura. And when you see the sputum of the patient, you can find what is called as dumbbell shaped asbestos bodies. There are other chemical carcinogens. The butter yellow that is used for taste, and there are some preservatives in it called the yellow of it, which can lead to malignancy. The plastic industries we had mentioned earlier, lot of these uses. In fact, plastic covers are being banned, but then there are the plastic floors, the plastic pipes, etc., which can lead to an angiosarcoma of the liver. Polyvinyl chloride is a substance. I would like you people to kindly by heart this list of carcinogens and the cancer that is being caused. Asbestos can lead to mesothelioma of the lung, bronchogenic carcinoma of the lung, and as well as mesothelioma of the peritoneum. Arsenic, skin cancer. Beryllium, lung cancer. Cadmium for prostate. Benzene, leukemia. Chromium for lung. Nickel, upper respiratory system or the lung. Vinyl chloride, angiosarcoma. Aflatoxin B1, hepatocellular carcinoma. Nitrates, gastric carcinoma. Aniline dyes, beta-naphthylamine for transitional cell carcinoma of the bladder. Please by heart this one. I would like you people very much to know this. Finally, a few words of wisdom. It is easy to kill the cancer cells. It is not as difficult as we think. But the challenge is at the same time, we have to keep the patient alive. So this is a specimen of the lung that is being infiltrated by the tumor. Imagine killing the cells alone, sparing the lung. And what is the meaning of life? Look at this particular contribution of the gentleman, Percival Pot. You find that the Pott's fracture is there. There is a Pott's tumor. Pott's cancer is again there. Pott's disease of the spine, etc. Lot of these are there, and I would like you people to kindly see his words. My lamp is almost extinguished. I hope it has burned for the benefit of others. It was in 1788, the last year before his death, he narrated these words, and it has remained a medical eponym. Gracias.